All right, welcome everybody. Thank you again for being here for today's webinar hosted by the Arkansas Native Plant Society. This webinar is being recorded. Afterwards, the recording will be uploaded to our YouTube channel. The mission of the Arkansas Native Plant Society is to promote the preservation, conservation, study, and enjoyment of the native plants of Arkansas. If you'd like to join, you can go to our website, amps.org/join. You can also check us out on Facebook, facebook.com/Arkansas Native Plant Society. If you have any general questions, feel free to give me an email at amps.president@gmail.com. All right, next two webinars coming up in our series on Wednesday, October 6th at 11 a.m. Dr. Travis Marsico from Arkansas State University at Jonesboro is going to talk about the role of herbaria and the recent discoveries of invasive species risks from hitchhiking propagules at international seaports. And then November 17th at noon, from noon to 1, it's a Wednesday, Jennifer Ogle is going to present for us on the history of the University of Arkansas herbarium. Our speaker today is Dr. Richard Abbott. Dr. Abbott is an assistant professor of biology at the University of Arkansas at Monticello, and he is also the curator of the university herbarium there. Dr. Abbott attended undergrad at Berea College in Kentucky, where he double majored in biology and German. Dr. Abbott then went on to study botany at the University of Florida in Gainesville, where he received his PhD. Today, Dr. Abbott is going to present for us on the native vines of Arkansas. Dr. Abbott, thank you for being here.
Cool. Thank you. Well, all welcome. Thank you for being here. So real quick here, what I want to start off by doing is if I can actually get out of this. This is strange. My computer. Oh, there we go. Nope. My computer is not letting me get out of. Here we go. I wanted to go just kind of very quickly, kind of just show you an overview of the presentation here. I wanted to kind of just share with you that my purpose here today actually is to kind of lay a foundation to help you learn how to organize information, how I go about organizing information with respect to plant ID. So the first 20 or 30 slides or so are basically going to be background information on how to think about organizing information. And then after that, there are a couple hundred slides, beautiful photos of all the vines in Arkansas, or not all, but most of the vines in Arkansas. There's no way that we're going to have time to get through all this. So I'm not even going to try, basically, is what I decided. I'm not even going to try to get through all these, but this presentation can be made available in PDF form. So we can talk about that afterwards, how to share this. But essentially, my goal here today is to introduce the information on how I think about doing plant identification, how I think about organizing vines in my mind. And then this presentation, once again, will be available to you so that you can come back and study the photos on your own time. But just be aware that we're not going to make it all the way through the presentation, through all the photos. There won't be time, but we'll get as far as we can. And so if there are any questions, I mean, unfortunately, I'm not quite literate enough with this to be able to interrupt myself and talk. But at some point towards the end, we're going to stop and ask for questions and hopefully have the last 10 or 15 minutes be y'all asking questions and me responding directly to any questions you may have. But for now, at least, let me go ahead and start talking about vines of Arkansas here. And precisely, just to be clear on this, is my approach to stuff is to think about what the words mean and well, what is Arkansas? What is a vine, basically? And trying to define things is always an important part of how I approach stuff. And so most of us are pretty clear on what we mean by Arkansas, right? I mean, we could obviously get into the kind of like etymological debate about why is it pronounced Kansas and why is it pronounced Arkansas, and you could talk about the British and the French and the Native Americans, and there's all that side to it. But the bottom line is that we're all pretty clear on what Arkansas is without having to define it, right? I'm not even going to attempt to define Arkansas other than just throwing up this map and saying, you all know what Arkansas is, right? And the same thing with vines, right? If I ask you, well, what is a vine, right? I mean, we all think we know what vines are, but the bottom line is if we actually started having discussion about most vine species, we would find some debate. And if you were to Google a definition search for vines, you'll find some definitions saying vines are woody plants in the grape family. And if you think about that, a vineyard literally means a yard full of vines, right? Ultimately, though, what exactly is a vine is not clear cut. There is no single definition. Vines are not a natural group. It's an artificial assemblage of things. And so just so that we're all on the same page, though, understand that what I mean by a vine is a plant that has a non-self-supporting stem. So plants that twine or climb or creep across the ground, you can call it clambering over the vegetation or coiling around things or kind of sprawling maybe. Usually though, these kind of sprawling prostrate herbs are not considered to be vines at all. Usually something like ponyfoot here, this Dichondra caroliniensis that we have here, is not considered to be a vine, even though it's in the morning glory family and all its relatives are vines, and maybe you would call it a vine, right? And so just be aware of this, that I'm not trying to include every possible herb with a viney habit. Some vines can actually form self-standing shrubs. I've found poison ivy as 5, 10, 15 foot tall shrubs all by itself. It doesn't stop them from being vines overall, though. And so ultimately, let's just be aware of this, that sometimes the definition of what is a vine is just not 100% agreed upon or understood by all, basically. And that's okay. That's part of life, right? It's a lack of clarity. And why is this not, for some reason, it's not advancing. Oh, there we go. And so Mitchella repens, this partridge berry is another good example here. Is it a vine? Is it a shrub? I know of one book on shrubs that includes partridge berry as a shrub. Most of us think of it as kind of an herb, this kind of low, prostrate, creeping herb. Is it actually a vine, though? Most people would not consider this to be a vine, and I don't. It's just this kind of creeping, prostrate habit. So just be aware. Once again, I'd like to kind of drive this home here, is that words like vine are actually just this kind of arbitrary point along a continuum of variation. There's no clear-cut line between what's a shrub, what's an herb, what's a vine. But in general, what we're going to include here are plants that have non-self-supporting stems, excluding some of these kind of prostrate, creeping herbs. And so, unfortunately, these Zoom settings here are blocking out the screen here, so I can't actually read my own screen here. Hopefully y'all are just seeing the slide here, but I don't. I actually have the top of my screen blocked out here by Zoom, which I can't make go away. So I think the number was 147. I can't actually read that. If I'm off by 1 or 2, I apologize again. I can't actually read what's in front of me because it's blocked. But in Arkansas, there are about 147 different species that are native and naturalized. And this includes 2 different basal angiosperms, 13 monocots, 130 something dicots. And the largest families of vines include things like the milkweeds, the dogbanes and milkweeds, right? The morning glories, the convolvulaceae, the squashes, the cucurbitaceae, and the legumes. The largest genera are things like smilax, the greenbriers have 11 species in Arkansas, clematis, the virgin's bowers or leather flowers have 9 species. And so if we're just talking about vines in kind of a global context, we use vines on a daily, weekly basis, maybe without even thinking about it, right? Vanilla, for instance, turns out is a vine. It's a viney orchid, right? So when you're using the flavoring called vanilla, unless it's the chemical derivative type, if it's wild vanilla, it's actually coming from the fruits of a viney orchid. Black pepper, which most people probably use on a daily basis, black pepper is also the fruits of a basal angiosperm. And so we have vines around us all the time, right here. And so if the goal in terms of figuring out vines was just to memorize a few random factoids like vanilla is a vine or black pepper is a vine, if that's all we were after was just a few random factoids, there's no way we can even begin to make sense of the Arkansas vines because there's going to be 147 different random species worth of factoids to memorize. So I much prefer myself an approach where we try to talk about the patterns and the groups that are there. And one of the values of this is if you want to be able to diagnose or identify something you've never seen before, understanding the patterns is a foundational step in that direction, right? And so there are these basic things that I'm going to talk about here the first few minutes here are imprinting versus diagnosing. Come back to that. It's key to understand the language of botany. There's no way I can talk about plants or vines without making sure that everyone understands the vocabulary involved. I focus when I'm teaching. I ask my students to focus on 5 basic features here. So we're going to talk about that, and then there's a few extra characters we use. And then being aware that when we're learning groups, when we're talking about groups, are they natural groups or artificial groups? And notice I've got quotes around that word artificial there. It's become kind of a norm, a mainstream norm among systemists these days, when we're talking about non-monophyletic artificial groups here, to put quotes around it to show this lack of monophyly here. So we're going to talk about a few of the artificial motifs. We're going to mention a couple of them in particular that I use. And then keep in mind that the last major kind of foundational block here is that we need to understand phylogeny. We need to understand relationships, because vines themselves are not actually a single natural lineage. Vines are an artificial human construct, right? They're biologically real organisms, but as far as a category, as far as something to talk about, this is just an arbitrary random group of things to talk about. And so we need to be aware of that. And I want us to all be like consciously aware of that. And so starting with a starting point here with this is talking about imprinting versus diagnosing. And I'm not going to spend a whole lot of time on this. If y'all would like me to come back and give another talk at some point going over this basic stuff, I would love to. But for now, at least, the goal is just kind of gloss over this stuff so that we can move forward and talk about the vines themselves. But what I mean by imprinting is the stuff that you've just kind of learned by rote memorization, stuff that someone just said, that's a cat, that's a dog, right? We all know night and day, stuff that you know without consciously thinking about it. That's what I mean by imprinting. So for instance, if we look at the picture over here to the left of the road, the sky, the feet there, if I ask you, is this person running or walking? If it has to be one or the other, right? We all know that that person is running. Why? Because when you're walking, one foot is on the ground at all times, right? When you're running, there's going to be periods of time when both feet are off the ground. There's so many words that we use. In fact, most of what we know, we've learned through imprinting, and we don't actually have the skills to diagnose what we think we know. But that's what education is about, right? That's why we go to college. That's why we take classes. That's why we study and learn, is to learn to actually diagnose what we think we know. And so, for instance, with the cat dog example, if I'd asked you, can you tell all cats and all dogs apart? Of course, right? I mean, no one makes it past the age of 5 or 10 in this, and at least in our society, without learning to recognize, without imprinting cats and dogs. But very few people can actually explain how to tell all cats from all dogs. And it turns out it's something as simple as the retractable claws of a cat. Dogs don't have retractable claws. But the question that I have for us then here is here I am talking about retractable claws. But have you ever had to look at a dog or cat's claws to know that it was a dog or a cat? Of course not, right? And this is the same thing that's going to happen with plants now. If you're already familiar with plants, you're going to look at some of the stuff that I talk about and think, why do I need to know that for? I can already tell these apart. I don't have to pay attention to this obscure feature. I can already tell those. But I'm going to back up and remind you that it's a big old world out there, right? There's a lot of stuff to learn. There's a lot of patterns here. And the last 30 years of my life have been spent studying and traveling and trying to figure out, well, what are these patterns? How can we boil it down to something that's a reliable motif, a reliable pattern? And so we're going to end up using some obscure things that don't make a whole lot of sense sometimes, except that they do once you understand the big picture context. So in talking with students, and I apologize, I feel like I'm talking really fast here. It's my nature, but I'm used to interacting more and actually asking for questions. And I want to ask, are there any questions right now? I do wish that I could ask that question, but of course, there's just no practical way of doing that right now. If you do have questions, please take note of them. Please follow up with me later. I love sharing my passion for plants with people. Be happy to answer questions. So hopefully you're following me. Hopefully I haven't said anything too confusing yet. We have 5 basic features that I ask students to focus on. The truth is, this slide that you're looking at right now, if you actually understood all the vocabulary here and knew how to apply it consistently and regularly, you would have the skills necessary to separate almost all plants. You would be able to walk up to almost any plant and figure out what group it's in, right? Now, of course, you still have to learn a couple hundred more vocabulary words in order to articulate what your brain sees. And then you have to deal with the thousands and thousands of names to put names on those groups. But ultimately, we're going to focus on these 5 basics for now, being aware that there's a few additional characters, right? So the 5 basics we're going to talk about are what are a plant's habit, right? Is it woody? Is it herbaceous? Is it a vine, tree, shrub? Of course, today is going to be primarily about vines, so we don't have to worry about habit today. What's the leaf composition? Is the leaf simple or compound? That is the blade dissected into leaflets or not? What is the leaf arrangement? Do you have 1 leaf per node? It's alternate. Do you have 2 or more leaves per node? Opposite. And what is the margin? Is it entire? Is it toothed? Is it lobed? And then these things called stipules. Stipules are paired outgrowths at the base of the petiole, right? And not all leaves have them, but when they do, it can be diagnostically important, right? So these are the 5 basics that I ask students to focus on, because with these 5 basics, almost all plants can be diagnosed to family, can be separated out, at least to an artificial motif, and when they have to use a few extra characters. But be aware, there are going to be a few times when flowers are necessary. It's absolutely sometimes critical to have flowers or fruits. So these 5 plant ID basics, once again, are just kind of a starting point, a foundational starting point here. And we're going to want to add a few extra additional characters. For instance, here in the lower left, where it says A, cuticular pits. Cuticular pits, the cuticle is that waxy deposit on the epidermis of a plant to prevent water loss, right? But it turns out if you put a bag over your breathing hole, you're going to suffocate, right? Same thing with plants. It turns out the cuticle is going to have little holes in it associated with the stomates. And for some reason in the olive family, the Oleaceae, these cuticular pits show up as little green specks on the underside of the leaf. I don't know why that's the only family that does that, but I've observed that almost all olives, almost all Oleaceae have noticeable, obvious cuticular pits. And you don't typically see that in other families, right? Great character. The Pavonulus B, we look over here in the lower right. The Pavonulus is that thick swelling at the base of a leaflet stalk, the base of a leaflet stalk. And if you look and you see how the Pavonulus is thick and discolored, and especially with respect to the adjacent midvein. The adjacent midvein is a different color and texture than the Pavonulus. That's a legume feature, right? That's a very nice distinctive thing saying, hey, I might have a legume. The milky sap that we see here in C is awesome. When you find milky sap, it's going to be a mulberry or a sapotacea or a handful of other things like a morning glory in terms of vines here, right? So milky sap turns out to be a very useful feature as well. Is there a sheathing leaf base like we see in D up here? Is there a sheathing leaf base? Monocots have a sheathing leaf base. Dicots normally don't, but it turns out that there are a few groups of dicots that can be characterized by having a sheathing base. So it turns out to be very useful. A feature like a tendril, especially is important then for vines. Only vines are going to have tendrils. Not all vines have tendrils, though. A tendril is a modified structure for climbing. When you find a tendril, awesome. It makes your life a lot easier to figure out what you're looking at, right? And so smell, venation, there are other features as well that can be used here. And so here we're looking at a close-up once again of different kinds of colored sap, different kinds of stipules. These are important features for identifying vines as well. If you find milky sap on a vine, you've just found a morning glory or a milkweed vine, right? It's one of those two families. If you find petiolar glands on a vine, these extrafloral nectar glands that you can see here to the right here, these petiolar glands are only on vines are only going to be found in passion flowers. Palmate venation, well, quite a few vines do that, so not super useful there, but it still can be useful though, right? And so this idea that we're going to use the 5 basics and then add a few extra characters from time to time is the foundation for organizing the information to put names on vines. I love flowers. They're really cool. I like flowers and fruits. What we're looking at here are flowers of a manihot, essentially the cassava genus, or yucca, if you've ever eaten yucca. Not yucca the monocot, but yucca, the cassava plant. Separate male and female flowers closely associated on the same plant is the spurge motif, right? The Euphorbiaceae family are characterized by that. But we've got one euphorb vine called noseburn, which and seeing its flowers are the only way to reliably 100% diagnose that genus or that group globally, right? But most of the time, we're not going to have flowers. Most of the time, most plants are not reproductive, right? You do not normally find flowers on fruits on most plants most of the time. So we're going to focus on vegetative features as much as possible. We're going to do our best to focus on vegetative features as much as possible. So when we start creating these motifs, then keep in mind that when we're talking about natural groups, a motif might be a natural group, or a motif might be some artificial assemblage that I've created because I think it's useful, right? So some of these motifs are going to represent natural groups. So if we look at monocots or dicots, those are natural groups. Those are groups of angiosperms, right? Anything with the quotes around it again is an artificial group here, is a group that I've made up, that I've put a name on because it's useful. This might seem overwhelming, this one slide here with all these words shoved onto it. But what you're looking on the slide here actually would empower you to put names on several tens of thousands of species with just these few words. And notice how it's most of the 5 basics, right? The blade, the veins, these kinds of basic characteristics that we're focusing on here. And so again, these motifs are very useful patterns for then trying to organize the diversity that is life, right? And so something like the word ochrea that was there, if you saw the Polygonaceae, the knotweed family, this is the family that includes the knotweeds, the smartweeds. It has the docks in it. If you've ever eaten rhubarb or sorrel, this is that family. Well, the Polygonaceae, at 1,200-1,300 species, not a huge family, but they almost all, there are always exceptions. There are always exceptions to these motifs, right? But it turns out that the stipules, those paired structures at the base of the petiole in many plants, are fused into a sheath in the Polygonaceae. So in the Polygonaceae here, if you see where the petiole comes down and hits the stem here, so we're looking at close-ups of stems here, close-ups of nodes. The basic growth module plant, right, is that you've got a stem, and the region of the stem where the leaf attaches is called a node. The first thing that attaches to the node is going to be the leaf itself, and there's going to be an axillated bud down inside there. Well, in the Polygonaceae family, in the knotweed family, those stipules that are there fuse into this sheath above the petiole. And that's cool. There's only a couple other things in the world that do that, basically. And so by learning to recognize this one word, ochrea, you've now got 1,000 species at your fingertips, right? Well, 700 or 800 at least at your fingertips. And so again, the motifs are intricately tied to the vocabulary. We have to know the vocabulary. We have to know how to apply it. And when we talk about the artificial groups, once again, I just wanted to kind of a brief aside here and talk about, we do not want our classification to be based on artificial groups, right? If we just looked at an animal that had wings and said, hey, that's pretty useful, that's pretty easy. I'm going to take bats and birds and butterflies and grasshoppers and stuff. I'm going to put them all together into a group because they have wings. We all know that's not a natural group, right? Because bats are mammals. Birds are what? The toothless, feathered remnants of the dinosaurs, right? And then these invertebrates, the things like butterflies and grasshoppers and stuff, are way different. So it's not a natural group, but would it be useful if you were just trying to put a name on something? Would it be useful to have a group called winged animals? Yeah, absolutely, absolutely. It would be a very easy way to do things, right? But we know that that's artificial. And so keep that in mind, that when I'm talking about artificial groups here, it's the equivalent of putting bats and birds and butterflies together. Don't think of it as classification. Think of it as a practical, useful way of putting names on something, not classifying. And so one of these artificial motifs that relates to vines directly is when a vine has a tendril at the node. This is the motif that I call tendril at node. So if you look in the very middle here, we start with the middle here. This is the Virginia creeper. This is a close-up of Virginia creeper, which is the grape family. And this tendril here, once again, the tendril, this modified structure for climbing or twining, is actually attached opposite the leaf. So we have the leaf down here at the bottom with the stipule there. The tendril is 180 degrees across the stem, across the node from the leaf. When you have a leaf opposed tendril, awesome. There's only one family in the world that does that, the grape family. So if you have tan, you can separate these three major families of tan just based on how the tendril is attached. Grapes have leaf opposed tendrils. If you look to the left here, this is Xerostichios. So I believe it's an African species here. If you notice, here's the petiole with the blade. The tendril is attached at a right angle to the petiole. If you have a right angle tendril, that's the squash family, the Cucurbitaceae. And if you look to the far right over here, this is a passion flower. Notice that the stem in the background, you got the petiole coming kind of curving down, and this reddish thing here, the tendril, it's an axillary tendril. Around here, if you find a vine with an axillary tendril, it's a passion flower. There are one or two other things in the world that you'd have to worry about, but around here, if you find a vine with a tendril with a node, it's going to be one of these three things, and you can separate them so easily. And this is the idea, once again, behind these motifs, this kind of foundational pattern for how are we going to learn to separate the vines? Well, by paying attention to characters like this and details like this. Okay, and I want to stress again this idea that we absolutely do not want to base our classification on artificial motifs. These motifs are great for ID, but they are not reflecting, when they're artificial, they are not reflecting natural variation. They're not reflecting natural lineages here. The flip side of that, and this is part of why I use artificial motifs, is that many natural groups are also not readily identified. There are lots of natural groups out there that have cryptic features that are hard to tell, that there's no known morphology for. The modern monocot families make almost no sense morphologically, a lot of them. If you've got DNA-based groupings, those DNA-based groupings are not necessarily going to help with practical field ID. Some of these groups can overlap or integrate with other groups, and then sometimes you have these false similarities or false differences. For instance, snakes. We know snakes are reptiles, but they don't have 4 legs. Well, snakes lost their legs. There's a false difference, though. Just because they lost their legs doesn't mean they're not reptiles. Just because birds have feathers doesn't mean they're not reptiles derived from the dinosaurs. And so understanding relationships is not always going to be predictive for ID purposes, but it usually is useful. Yes. So often you do want to pay attention to relationships, but you have to know when to focus on a natural group versus an artificial group. So I wish I could stop for breathing here, but Eric, I'm just wanting to verify that everything's going well. Any questions or problems? Any reason why I need to stop or slow down for a second?
I think you're fine. I mean, if anyone has any questions, just put them in the chat. I think you're doing great.
Okay, cool. Thank you. Yeah, I'm unused to talking without the interaction, so I apologize for asking that question, but thank you for letting me know that. All right. So now what we're doing here is we're looking back again at this kind of phylogeny. This cladogram is a representation of relationships, right? We're showing sister group relationships here. We're showing what's most closely related to what. And so we have three major groups of angiosperms. And angiosperms are these plants that have flowers, fruits, double fertilization, new and improved vascular tissue. There's several things they have going for them basically that make them different from earlier plants. And there's three major groups of angiosperms: monocots and dicots, most of us have heard of. But then we have these lineages, these basal lineages, the early lineages of flowering plants that we're still trying to figure out, what does it mean to be a flowering plant? What does it mean to have flowers? What does it mean to have fruits? And so some of these basal angiosperms are going to look more like monocots, some are going to look more like dicots, and some are just going to be their own weird thing, doing their own thing, and look nothing like anything else. I'm going to talk about them as a group, not because it's a natural group, because it's useful. It's a practical, useful way of dealing with these unrelated lineages of early derived angiosperms. So when we talk about something like monocot versus dicot, just very quickly to review the basics that I'm sure most of you are familiar with here, is that dicots have broad blades. The blade is the flattened photosynthetic portion of a leaf here. Broad blades, reticulate veins, these branching net-like veins, and the leaf base is usually not sheathing. In contrast, a monocot has a linear blade, a long line-shaped blade, parallel veins, and a sheathing base. Same thing here. You can see the parallel veins, the reticulate veins. There's different types of reticulate veins, right? They can be pinnate, they can be palmate, they can be arcuate. There's different kinds of veins. There's lots of different vocabulary terms out there. But fundamentally, if you see a plant with a linear leaf, linear blade and parallel veins, it's going to be a monocot. A broad blade with reticulate veins is going to be a dicot. And so these are actually natural groups that are also useful motifs. So if you want in the future, I'd be happy to go back and talk about this foundational stuff more clearly. But for now, we're going to move on from that. But just be aware that once again, monocots and dicots are natural groups that are also motifs. But I asked this question here: are they invariant? The answer is no. Of course they vary, right? There are going to be monocots that don't look like monocots and dicots that don't look like dicots. There's always variation, always exceptions. But we're going to focus on the foundational norms for now. And so the first step for me then, when I start then trying to say, okay, well, what are the vines of Arkansas? Right? This is the first question in my mind. I'm going to talk about the vines of Arkansas. But what are the vines of Arkansas, right? Vines it's an artificial group. And so since it's an artificial group, there's no set criteria for what it takes to be a vine, right? I have to figure out what I'm going to include in this talk, basically. And so the first thing I did is I sat down, I made a list of things that I know to be vines in Arkansas based on my training. And so this list here is not every vine in Arkansas, but it's most. It has most of the vines here, just to illustrate that for me, the first starting point to defining an artificial motif. How do you go about creating an artificial motif? Is you have to figure out what species you want to include into it, what families, what genera, what do you want to include in your motif, right? And once you've done that, you can take those basic features like having compound leaves. And so, okay, of the vines of the vines in Arkansas, these are the only ones with compound leaves. Of the vines in Arkansas, these are the only ones with opposite leaves. Of the vines in Arkansas, these are the ones that are alternate, simple, and entire. And so by creating a few artificial motifs like this, we're able to pull out the 147 species and start winning away at it. And then I can create some artificial motifs like tan, these tendrils of the node, and pull out a few more species. And this is how essentially we end up breaking down the diversity, right? And so this idea of that old saying about how do you eat an elephant, one bite at a time, right? And so when you've got 300,000 species of plants in the world, 20,000 in North America, a typical state's got 3,000 species, you've got 16,000 genera, 150 to 200 families in a state. It seems overwhelming, right? But if you can stop and think about, well, what are the natural groups, right? What angiosperms, monocots, dicots, cool. We can break them down that way. Orders are groups of related families, right? We can talk about the buttercup order, which includes the moon seeds. The sapindales includes things like poison ivy and also balloon flower. Solanales includes vines like the morning glories. Lamiales includes vines like what? Trumpet creeper, the bignoniaceae families. Then you got families. You can think about the families themselves. Well, how do you identify the squash family, the grape family, the passion flower family here? Well, we've already mentioned that. But this is the idea, right? Understand what the groups are, and then you can figure out how to actually then diagnose them. And that's the goal, right? Figure out how to put names on all these things. This slide is a reference for those of y'all that want to start trying to build a better foundation. There are lots of cool websites and resources out there that provide information for understanding the bigger picture groups. If you want to understand about the orders and the larger groups, there are websites for that. So this is just some online resources that I recommend you look into if you want to try to learn this kind of higher level stuff. For our purposes, though, what I want to start doing now is working our way back into the actual vines and actually maybe talking about a few of the vines here. So just going to build again, just kind of quickly go back over the foundation here. We've got this group called basal angiosperms. It turns out that basal angiosperms as a motif can be characterized by being woody, simple, alternate, entire, no stipules, and have a strong, spicy smell. There are, of course, going to be exceptions to all of these things, right? There are a few basal angiosperms that are herbaceous. I don't know if any that are compound, but there are some that are going to be opposite. There's a few that are going to be toothed, and there's one group of basal angiosperms that have stipules, the magnolias, right? And then some basal angiosperms have no smell. So there are always exceptions here, right? But the bottom line is that this is still a useful motif because 80 or 90% of the time, if you walk up to a plant that is woody, simple, alternate, entire, no stipules, and has a strong, spicy smell, it will turn out to be a basal angiosperm. So this is a very useful way of dealing with most of these angiosperms that are not monocots or dicots. Just an artificial way of saying, hey, let's put a name on these things that are not monocots or dicots. And the basal angiosperms for us, just to make this something that you might be familiar with, things like magnolias and sassafras and water lilies and pawpaws, star anise, and the amberella is this thing over in New Caledonia. It's a very different thing. But these are the examples of basal angiosperms that we have around here with a couple others thrown in. And so getting back on track, what the rest of the talk here basically is going to be is looking at some pictures and talking about some of these groups here. We have one fern. It's an invasive fern, the Japanese climbing fern. We have two species of basal angiosperms here, with the Dutchman's pipes and this thing called Schizandra. We have 13 different monocots. 11 of them are greenbriers, the genus Smilax. 2 of them are yams in the genus Dioscorea. The vast majority of our vines in Arkansas are going to be dicots, right? But it turns out that roughly, a fourth or a fifth of those, a fourth or so of those have opposite leaves. Okay, let's pull those off first. Let's pull off the opposite ones. About 51, almost half of them have as far as the species go, have compound leaves. This is it. Let's pull those off, right? There are only 3 genera that are going to be compound and opposite. So by putting 2 characters together now we've narrowed ourselves down to being one of 3 things here, right? So there's this really useful way of using this kind of conceptual motif-based framework and a few basic vocabulary words to separate all these vines apart so that you then have a framework for learning vines and learning how to diagnose vines. But what this leaves us with is about 55 species of vines that are going to be what? They're going to be alternate, they're going to be simple. So we pull off again the compound opposite, pull off the compound alternate, pull off the simple opposite things here, pull off the ones, and then you can separate those based on whether they're entire or toothed, right? The entire opposite thing is going to be the milkweeds, Gelsemium, the Carolina jessamine or yellow jasmine, and the honeysuckles. The ones that are toothed, things like hops, our climbing hydrangea, Kixia, these invasive things, or Mycania, our hemp vine, right? What it boils down to here is that roughly half of our eudicot vine species, roughly half of all the vine species are these eudicots that have alternate simple leaves. Now, if you don't know how to tell a basal angiosperm or monocot, you could obviously throw those in here as well, right? There's no reason why you can't modify this motif to include basal angiosperms and monocots. You just have to add them in where they're appropriate here. But what we've done here now is with a few basic framework terms, with a few basic terminology, a little bit of an understanding of relationships here, we've now narrowed ourselves down to just these last few groups here, right? So how do we go about separating these last few groups? Well, it turns out a third of our dicot vines have tendrils at the node, right? About a third of our species are either going to be grapes, passion flowers, or squashes. There's a third of them. Half of our species of dicot vines are going to be entire. So we look at this and we're like, okay, well, this is the knotweeds, the buckwheat vine. About 30 of those are actually morning glories themselves. Morning glories have milky sap. So boom, once you know how to recognize that a vine is simple and alternate with milky sap, you've got a morning glory. So roughly a fourth of all the vines in Arkansas are morning glories, right? 30 out of 147 or so. So a fifth, I guess a fifth is a better number there. But if you've got a vine, once again, that is simple, that is alternate with milky sap, that is a morning glory. And so this is the idea behind these motifs. Once again, this framework is use 2 or 3 basic, simple ID characteristics to pull apart all the species. And what we're left with here is the dicots that have simple alternate leaves here that are then toothed and or lobed. There's just 5 things left, right? The moon seeds. Well, they've got these twisted kinked petioles, no stipules, they're lobed with no teeth. Sometimes they're not actually lobed. Sometimes you find cocculus with no lobes at all. The bittersweets, they're pretty nondescript, right? The celastruses, they've got low teeth, rounded to obtuse bases. They've got stipules. So part of what makes the bittersweet so distinctive is their lack of distinctiveness, if you will. It sounds kind of crazy to say, but the bittersweets don't have the distinctive features that allow you to recognize these other groups here. Our one native viney noseburn, the trachea that we have here, has cordate leaves. It's very densely hairy. It has jagged teeth. It has stipules. It's got separate mesenteric flowers closely associated because it's a spurge, a euphorb. Rattan vine, Berchemia scandens, right, has these pronounced parallel secondary veins. There's usually 6 or more. Most of these other vines are only going to have like 4 or 5 prominent lateral veins, and then the stipules that are present. And then English ivy, right, has a slightly sheathing petiole base. It's got a smell to it. If you crush up a leaf, it has no stipules. It's evergreen. So here we go. Once again, we've now used just a few basic terms, an understanding of relationships, and what we've just done is essentially separated all of the vines of Arkansas into little knowable, discrete chunks, right? We've just eaten this elephant in lots of little tiny bites. There's one thing to be familiar with the terminology, to have the theoretical knowledge. It's a second thing to actually have the applied hands-on skill. And that's where being out in the field and becomes so important here. Because the theory, this idea that we've got these basal angiosperms, monocots, and dicots, is just a framework for reminding yourself, well, what does it mean to be a basal angiosperm? What does it mean to be a monocot or a dicot? What does it mean to be these different groups? What does it mean to be a grape? Well, a leaf opposed tendril, right? What does it mean to be a moonseed? You got to have those twisted, kinky petioles, those kind of kinked up, twisted petioles, right? So ultimately, what it boils down to here is just trying to understand and know what these different words mean and what words to use as their motif, right? And so moving on then with this last little bit of time that we got here, the next 15, 20 minutes or so here, let's go ahead and flip through some of these pictures very quickly. Once again, we do not have time to explore all the vines of Arkansas. 147 species would take us a few hours to work through, basically. What I'm going to do is just kind of skim through this stuff fairly quickly. This presentation is available as a PDF once we can figure out how to share that with y'all. So you can come back and study this on your own here. But basically, remember this, we've got 2 basal angiosperm vines, Schizandra and Dutchman's pipe. These are basal angiosperms, which means what? They actually have a strong, spicy smell to them. So if you walk up to a vine and you crush up a leaf and there's a strong, spicy smell, you've probably just found Schizandra or Dutchman's pipe, assuming the leaves are simple, right? I'm going to give you one kind of spoiler. Poison ivy also has a strong, spicy smell, right? You don't want to crush up poison ivy. Poison ivy has compound leaves. So if you walk up to a simple leaf vine, though, you're going to be safe at crushing it up and smelling it. If it's a compound leaf vine, don't crush it up until you know it's not poison ivy. Monocots, we've only got 2 groups of monocot vines, greenbriers and yams, right? And so we're going to talk about those briefly. And then the dicots, once again, this is kind of an overview of just showing how the relationships within the dicots here. These common names that you see here, things like clematis, moonseed, knotweed, are going to be represented by the scientific names in the photos. So if you're not real experienced with matching up scientific names with common names, and you have a question, please contact me, because the photos are going to be labeled again with the scientific names, not with these common names that I'm using here. But understanding that vines, once again, when I say that they are not a natural group, it's an artificial assemblage. This cladogram is pictorially representing that, showing us that what we're calling vines are just an artificial group of things that are not related to each other at all. And the last major group of vines here within the asterids, this group called asterids here, the sympetalae, these are the plants that have fused petals. These are the plants that have the petals fused to form some sort of corolla tube here, and includes things like the milkweeds and the hemp vine and honeysuckles. All right. So again, I wish I could stop and ask for questions right now, and in fact, I'm tempted to. And so Eric, what are your thoughts on this? I've essentially covered the foundational stuff now, and the rest of this is just photos. I would love to open the floor up to questions about specific groups, or would you rather I just spend another 15, 20 minutes going through some of the pictures?
Let's go through some of the pictures. I think a lot of people have been waiting on that part. Okay. So let's do that, and then we can have photos at the end. Let's just spend the next 15, 20 minutes going through some of these pictures.
Awesome. Cool. Thank you. Thank you.
Thank you.
All right. So this first here, the invasive Japanese climbing vine, right? It's a horrible, invasive, nasty weed. Dutchman's pipe, Aristolochi, also now called Isotrema by some. The Dutchman's pipe here, basal angiosperm, crush up the leaf. It's going to have a strong smell to it. It's got a cordate leaf base. It's simple. It's alternate. It's entire. You're looking at, there's no stipules on it. It's got palmate veins. And so the features that are here, the vocabulary that's here, is what you need to be consciously articulating to recognize that you've got a Dutchman's pipe. If you've got flowers or fruits, unmistakable at that point in time, right? Really cool, weird trap flowers, the really bizarre backwards opening capsules that hang there like little baskets that the seeds fall out of. Really cool, weird flowers and fruits on the Dutchman's pipes. The other baseline angiosperm is the one Arkansas vine I've never seen. I've never seen Schizandra glaber yet. It's only known from a few counties of kind of central eastern Arkansas, and I've just never seen this. In 30 years of botanizing, I've never seen it. I'm going to assume that if you crush up a leaf, it's going to have a strong spicy smell because it's a baseline angiosperm. I'm looking at these photos here, and I can see that it's woody, simple, alternate. I was going to say entire, but this actually looks toothed. Schizandra looks like it might be one of the baseline angiosperms that has teeth. That's kind of cool. So that's what would make Schizandra different from the rest of the baseline angiosperms, is having teeth. Almost no baseline angiosperms have teeth. You can see here it talks about male and female flowers. That's kind of unusual. Not too many baseline angiosperms do that. Having separate male and female flowers is kind of unusual as well. So anyway, this is one of our rarest of our native vines here, and it's one that I've never seen. I borrowed these photos from the internet. The yams, the wild yams. We have one native wild yam called Dioscorea villosa. People in the past have tried to call it two different things, and there are people who still are convinced that there's two or more species. But the evidence is pretty overwhelming that this is just one widespread species complex, which may or may not have some distinctive populations out there. And so this native wild yam, if the goal was to try to teach you how to diagnose and recognize all the species, I need about 10 hours basically to work through all these things basically. So I'm just going to gloss over these quickly here, but I'm going to try to draw your eye to some of the attention, some of the key details here. If you look, the cordate leaf bases, notice those veins, how they're strong, how they're palmate at the base, but the lateral veins are not coming all the way to the tip. The lateral veins are not extending all the way to the tip of the leaf, right? They're only going part way up. Unlike the greenbriers, where the lateral veins go all the way up to the apex. The yams are unusual in monocots and having separate male and female flowers. So you have these racemes, or sometimes panicles, with separate male and female flowers here with an inferior ovary, which sets them apart from most other monocots, but not all. Very distinctive fruits in the yams. These capsules that open up with these flat winged seeds. We have an invasive yam as well. The Chinese yam, when I first learned it, it was Dioscorea cinnamomea. Then it became Dioscorea oppositifolia. Now it's being called Dioscorea polystachya. Notice once again, those lateral veins are not actually making it all the way up to the margin, which separates this from the genus Smilax. Notice that there are no stipules. There are no tendrils, which is also going to separate it from the genus Smilax. This species is differentiable from our native one because it's got these lobes to the leaves. And you can actually find opposite leaves on this. In our native species, sometimes the lowest node or two or three or four will have whorled leaves, but having opposite leaves distally on the plant, our native yam doesn't do that. The Chinese yam does. This species also sometimes forms little bulbils in the axils. Our native greenbriers, we've actually got 11 different greenbrier species. About half of them are woody with prickles, the other half are herbaceous. I'm not going to try to talk about them in detail. I could spend an entire hour just trying to teach you about the different Smilax species. I'd love to come back and talk about them. But what I'm going to do is just kind of flip through some of the pictures very quickly. Once again, what we're looking at to recognize that a Smilax is a Smilax, it's the only genus in the world that does what? Has paired stipular tendrils. So either one of these pictures you look at here, you can see the leaf blade, the petiole down at the base, it actually at the sheath there, we've got these paired structures, these paired tendrils coming off of the petiole. Here's the leaf. Paired structures coming off of the petiole there. Paired stipular tendrils. If you find a vine with paired stipular tendrils, it's a Smilax. The only genus in the world that does that, right? So it's a very easy genus to recognize when the tendrils are present. They also have separate male and female flowers, and instead of having elongated racemes, the inflorescences of the greenbriers are umbels. All the flowers are radiating from a single point like this. And instead of having the inferior ovary of the yams, here's the ovary here, this bright green structure here is the ovary. There's the stigmas. Here's the tepals down here. This is the superior ovary. So they're different in a lot of ways, basically. Our glaucous greenbrier has that bluish whitish coating on the inside leaf that rubs off. The bristly greenbrier will have these kind of minutely erosed margins here. There's these little tiny projections here, and it often has the pandurate leaves. At the base of the plant, at the base of Smilax tamnoides, the bristly greenbrier, which I'm seeing some sources are now going back to calling this Smilax hispid again. It has these very narrow needle-like bristles, these dark purple bristles. But that margin of the bristly greenbrier, once again, is minutely erosed. These little pegs along the margin like that. One or two of our other greenbriers can do that, but most of our greenbriers are not going to have those little minutely erosed margins. Smilax rotundifolia, if you look at this one, it's also going to have a minutely erosed margin, strongly angled stem. The petioles are often red in this species, that minutely erosed margin. And look at the prickles here, these sharp, pokey epidermal outgrowths. See how they kind of have 2 or 3 different colors on them, and they're very broad at the base. Very different prickles than what you find in the bristly greenbrier, right? So training your eye to see things like the minutely erosed margin and the different types of prickles is how you separate the Smilax species. Smilax bononox has bristles along the margin. It's a very bristly species. It's almost always going to have green petioles. It's very rare that it would actually have red petioles. But the bristly margin, when the bristly margin is not present, it will be entire. It will not have those minute erosions. It will be a completely smooth entire margin when the bristles are not present. The herbaceous smilaxes, I'm not even going to try to separate those. They're tough. They're really hard. These herbaceous smilaxes look a lot like the Dioscorea, right? The Dioscorea villosa. But notice how the secondary veins are going all the way to the margin, all the way to the apex. The secondary veins are rejoining all the way up at the apex, unlike what you find in Dioscorea. And so the herbaceous smilaxes are questionable how many species there actually are, but they differ in some subtle ways. And it's entirely probable that we have not accurately described them in the landscape. There's definitely some images on iNaturalist which don't quite fit anything out there. But our herbaceous smilaxes all have the same basic look to them, right? They're not going to have prickles. Sometimes they'll have the tendrils, but sometimes they don't have the tendrils. How you then separate them from the Dioscorea when they don't have the tendrils is once again the secondary veins. I'm sorry, the primary veins, the lateral primary veins are extending all the way to the apex in the greenbriers, but not in Dioscorea. Our second largest genus of vines here then is the genus Clematis. There's 2 major subgenera of Clematis. They're called Virgensbauer when they have white petals, sometimes yellowish, that have rotate symmetry here. And then when the tepals or petals here, you know, are purplish or bluish, and they're kind of coming together and then forming like a bell or an urn shape. These are referred to as leather flowers. So we have 9 different species of Clematis in the state here. Common ones like this. The white flowered ones, Clematis virginiana type, is the most common one that I've seen so far. So the typical flowers of these species. Here's the invasive one, Clematis terniflora. Notice how it's got kind of entire margins. And so here it is up close here. So Clematises have opposite compound leaves, right? So this is a compound leaf. This is a compound leaf, and they're opposite each other. The only other thing in Arkansas that you're going to confuse a clematis with, the only other vine that has opposite compound leaves, are the 2 genera of Bignoniaceae. And you can separate Clematis from Bignoniaceae if you look carefully here at the node. There's a nodal line, a nodal ridge in Clematis that you will not find in Bignoniaceae. If you look at the petioles, the petiole, when it comes up where it hits the blade, where it hits the leaflets, above that's then referred to as petiolules. So the petiolules, the stalks of the leaflets, notice how the petioles are twisting and curving and twining as if they were tendrils. They're not tendrils. It's just petiolules acting like tendrils. Well, Bignoniaceae don't do that. Bignoniaceae will either have tendrils, real tendrils, or won't have anything like tendrils at all. But they will not have petiolules twining like this. So you can easily separate Clematis from the Bignoniaceae. And why did I put them together again? Because they have opposite compound leaves. So again, more Clematis. More Clematis. Case beyond this is more of a northern one in Arkansas, which has the biternate leaves. And here it is in fruit here. This is the plumose style. This is actually the remains of the female part of the flower here with the achene there at the base here. Again, love to go back through all these families more slowly in the future if you want to have a follow-up talk. But for now, I'm just going to keep rushing through these things. Now we're getting into some of the purple flowered ones of the Clematis crispa type here. You can see once again that nodal ridge is still there. You can see the petiolules are still acting like tendrils. And here we have the typical kind of purple bell-shaped flowers that you find in this leather flower subgenus. The Clematis crispa is the most common one that I personally have seen so far in Arkansas. I've only been in Arkansas. I'm starting my 4th year now. Here's an erect. I threw this one in from Florida just to remind us that a few Clematises are actually erect herbs. There are a few species of Clematis that are not actually vines at all, but not in Arkansas. But you got to go elsewhere to see that. So related to clematis, related to the buttercup in the ranunculales, right? We have this family called the moonseed family, the Minispermaceae. We have 3 genera and with 3 species of moon seeds in Arkansas. And what do they have in common? They have these twisted, kinked petioles. So here's the blade, the big flat expanded photosynthetic blade here with the palmate veins. And notice how twisted that petiole is. That twisted, kinked petiole characterizes the moon seeds. The only other vine that you're going to see around that looks like this that also does that is the Dutchman's pipe. And how would you tell that apart? Crush it up and smell it. The Dutchman's pipe is going to have kind of a strong, spicy, kind of a harsh, fetid smell to it. You won't get much of a smell out of a moon seed. So the moon seed, Cocculus carolina, the Carolina moon seed itself can have highly variable leaves. These are all the same species. So look, they can almost be like star-shaped, or they can be pretty much entire here. The flowers of these moon seeds actually have 3 tepals. That's weird. That's a monocot feature, right? So here is an early derived eudicot that has monocot-like flowers. That's kind of cool and weird. The seeds, the reason they're called moon seeds is, so here we are looking at the fruits of this cocculus. These kind of strange half crescent moon seeds characterize the family, the Minispermaceae. And then Minispermum canadensis is another common one. And it's once again, you can see that it's got the kind of the kink to the petioles here. None of these photos show that real well here. This kind of slight twisting or kinking to the petioles. In this case here, it's also got peltate blades. The petiole doesn't attach at the margin. The petiole actually attaches into the blade a little bit, referred to as peltate. So it's a very distinctive species. And Calicocarpum looks like kind of a mix between this and the cocculus. And so I threw in a couple vines here that are not known from Arkansas yet. One called chocolate vine, Achevia quinatus, and the Lardizabalaceae, which I like saying that family name. My major professor in grad school, Dr. Walter Judd, said that the graduate students at Harvard, so this, Harvard of all places, for the Lardizabalaceae, they said, "Lord, give me a lizard ACE." So this is the Lord, give me a lizard ACE family. And so the Lardizabalaceae, the chocolate vine, is a cultivated plant which is becoming invasive all around Arkansas. And it's just a matter of time until someone finds it here, because it is being cultivated, it is being sold in Arkansas, and it has shown itself to be invasive elsewhere. And it's going to be one of the very few vines that we have that actually has palmately compound leaves like Virginia creeper. All right, this ochrea that we already learned earlier characterizes what family? The knotweed family, the Polygonaceae, right? So once you've learned to recognize this ochrea, this stipular sheath, this fused stipular sheath here, you've recognized this family. So in this family, we have something called buckwheat vine, Berniccia ovata. Buckwheat vine looks like it has tendrils. It looks like tendrils, but it's actually just a twining stem. It's just a stem itself that twines. But when it's a stem that's the lateral branch, when it's an axillary branch that's twining like a tendril, it's going to look like a tendril. But ultimately, it's not actually really a tendril. It's just a tendril-like branch. And if you look carefully at the picture here in the middle, you can see this kind of ridge around the node there. That's the remains. That's the vestige of the ochrea. The ochrea in buckwheat vine is very reduced, very small and hard to see sometimes. The fruits of buckwheat vine are really bizarre and cool. These are these strange achenes, right? We've got this kind of flattened winged lower portion here. The remains of the tepals up top there. Very bizarre, cool, weird fruits in our native Berniccia ovata buckwheat vine, which is a member of the Polygonaceae once again. I threw this one in, Persicaria scandens. There's several Persicarias that are kind of scandent. They're kind of sort of viney. Are they herbs? Are they vines? Well, I don't really call this one a vine, right? This is a group, the Segret group that used to be called Tracholon, used to all be called Polygonum. They're kind of sort of viney, right? They're kind of sort of viney. So I'm mentioning them. They're not really truly vines though. They're just kind of sprawling herbaceous things that have slightly viney habit here. And this group tends to have these retorse hairs on the stems, so they're very grabby. If you rub up against one, it's going to stick to you. Related to these as well, we've got this genus called Falloppia. It used to be Polygonum again, but the clade called Falloppia now. These buckwheat vines, essentially related to knotweeds. These knotweed vines, the genus Falloppia. And once again, you can see the ochrea down there. You can see the achene that characterizes the Polygonaceae. But learning to recognize the ochrea allows you to put recognize this family basically. And so here's another one of the Falloppias here. And now we're up to the grape family, right? So now we're getting to a family that's actually got several species, lots of species in the state here, several genera here. And this is a photo I took where I put Ampelopsis and Parthenocissus invitus together in the same photo, just to compare and contrast. The grape family can be simple, can be palmately compound, can be pinnately compound, or even twice compound. What do they all share in common? Leaf opposed tendrils, tendrils opposite the leaves. So once you've learned to pay attention to the fact that there is a tendril and where it's attached, it doesn't matter how different they look from each other. They're all members of the grape family. All right, so I'm going to just run through these very quickly. We're almost out of time here, so I'm just going to kind of flip through some pictures very quickly here. Some grad school buddies there, Kurt and Barbara here. Old grapevines, most of y'all have seen these around, right? They're out there, they're all over the landscape. They're super common out there. Lots of grapes. The summer grape, Vitis estivalis, with a leaf opposed tendril there. Summer grapes, super variable, highly variable leaves in some of the grapes. You've got to learn what characters matter and what characters don't matter. The gray grape, the Vitis cinerea here. More Vitis cinerea here, the gray grape. The Vitis labrusca. This is one that's not yet documented for Arkansas, but might be. It's in all the states around Arkansas, so it might be here somewhere. The muscadine grape. Again, if you want to schedule a follow-up to talk about how to separate the species, happy to do that. But for now, just kind of showing you some pictures here. Muscadine grape again. And again, the top of my screen is blocked out. So I'm just going to kind of scroll through these pictures right here pretty quickly here. Raccoon grape, Ampelopsis, different genus. There's our Virginia creeper again with the palmated compound leaves. There's actually a second Virginia creeper species out there. See the sucker discs that form on this? Parthenocissus quinquifolia forms sucker discs. The other Parthenocissus inserta, which is a more northern and western species, but may or may not be in Arkansas. I don't remember seeing it mapped for Arkansas. Doesn't have the sucker discs, but it might be out there. The thing is, you have to be aware of it and look for it to find something like that, right? Close up of the flowers of Virginia creeper here. And so at this point in time, I mean, again, there's no way I'm going to possibly make it through all the rest of the photos here. If we stop and look for a second here, I'm on slide 105. There's 146 more slides to go, right? No way we're going to make it through all these families. So what I'll do is just kind of stop here. And if there are any questions, if y'all want to funnel questions through the chat room, I'm happy to talk about other families, happy to answer any questions. But then if you want to follow up by looking through these other pictures yourself and follow up with questions, that's what these other slides are for, is for you to kind of study on your own and follow up with questions if you have them. But at this point in time, I'm just going to kind of jump to the end here. And my final slides here is just basically just saying, hey, I work with the Polygalaceae. This is me, 20, 25, 30 years ago in Mexico. The Polygalaceae family, the milkworts around here, most of the genera of Polygalaceae are actually liana, actually woody vines out there. There's a lot of really cool, weird vines out there. And the bottom line is if you want to learn to identify them, know the natural groups, learn the artificial groups. And I thank y'all very much for your time.
Yeah, thank you. Thank you, Dr. Abbott. That was a great presentation. Really enjoyed that. We got some great feedback here in the chat. Want to encourage people to go ahead and if you have any questions, enter them into the chat. Just want to read to you a couple of the feedback that we've received. Ellie, I'm assuming Ellie Polzer maybe from the Mississippi Native Plant Society. Says your passion for teaching botany is evident. I specifically enjoy how you frame and summarize the basics upon which to build. I'm here in Mississippi, and it would be great to be able to share this PowerPoint with our Native Plant Society if possible. Thanks so much. And I will tell you that Dr. Abbott did send me a copy of that PowerPoint. I'm sure he would be happy to send it to you if you reach out to him directly, but I'm also happy to send you a copy as well if you want to send an email to anps.president@gmail.com. Karen Seal, who is with the Arkansas Native Plant Society, says, Dr. Abbott, as someone who has never had a botany course except for what I learned in my 10th grade biology class, this presentation has been the single most helpful one I've seen in my 25 plus years of attending Arkansas Native Plant Society meetings. Thank you so very, very much. So I think, it's safe to say that, your presentation has been very well received. I think there's a lot of great information, and we would love to have you back to present, in 2022. I'm currently working on the program for that. So even just like a presentation teaching people how to separate the different grape species would, be great, or some of the larger families, of vines or other families here in Arkansas.
Yep.
Let me look here in the chat. Some other great feedback. Rose Tacker did ask if anyone knows, or if you know where she can buy muscadine vine. I know it seems to grow a lot in southern Arkansas and southwest Arkansas.
Yeah, I don't know the native plant nurseries well enough to know where might be selling them, but some native plant nursery might have them, but I don't honestly know in Arkansas.
If you go to the Native Plant Society website, one of our pages on there does have a list of native plant nurseries in Arkansas. Also, the Wild Ones website, the Ozark chapter, if you go to ozark.wildones.org, there's also a list of the native plant nurseries in Arkansas and surrounding states on that site as well. I just encourage you to reach out to some of them and see if they happen to carry muscadine. You might also reach out to Mary Ann King with Pine Ridge Gardens in London, Arkansas. She may or may not have some specimens to sell. And LifeQuest, I'm not sure who that is, but they mentioned the Audubon Society is having a native plant sale. So you might, that's down there in Central Arkansas. So you could reach out to them to see if they have any muscadines in their inventory that they're selling. Matt asks, are there many edible vines? They know that muscadine is edible, but are there other grapes edible?
Yeah, all of the grapes are edible. None of them are toxic or poisonous. None of them are going to hurt you to eat them. Most of them aren't very palatable, though. I don't even find muscadine to be all that palatable, to be honest. But yeah, all the grapes are going to be edible. I'm trying to think other vines. Yeah, there are lots of vines that we eat, like kiwis. Kiwi fruits are actually grown on vines. So rose hips, some of the viney roses, you can actually, you can frequently eat the rose hips, or at least make a drink out of them or whatever. I'm just trying to think. There's some stuff like moon seed. I wouldn't recommend eating like a moon seed, because the ranunculates, the buttercups and relatives, tend to have some very toxic carcinogenic chemicals in them. So I would definitely not just randomly eat any fleshy fruited vine. But off the top of my head, I'm not, no other like super edible vines in Arkansas are coming to mind. But if we were to look back through it, the passion flowers, for instance, that we have in Arkansas are not very tasty, right? The maypops is not a very tasty, but passion fruits are super tasty, right? I mean, if you get the edible commercially grown passion fruits, those are delicious. And of course, the squashes, all of the native squashes are technically going to be edible. They're not going to be in the sense of not being poisonous, but most of them aren't very palatable, right? So there's a difference between being edible and palatable, and most of the things are edible. They're just not very palatable.
Yeah, that's very true. And it looks like Sarah and Diana Soteropoulos also bring up a good point that Smilax shoots. The fresh growth on those is edible. Tastes pretty, compared to like a proteiny type flavor, but the new growth on that before it gets too woody, is supposed to be pretty high in protein as well.
I would caution against Smilax just for one. There's a study that I saw that suggested some carcinogenic compounds in Smilax.
Oh, really?
The thing is, when they force feed a rat the equivalent of 10,000 times what you would ever eat in your lifetime. So yeah, right. If you force feed a rat 10,000 times what you'd eat in your whole lifetime in a day, they might cause cancer. So personally, I would be careful with Smilax just because they might be carcinogenic.
That's good to know. Yeah, I didn't realize that. Sarah Gertz also says maybe Starks Brothers for muscadine. I'm not sure where they're located, but she might be able to provide more information. And you may have also answered Matt's other question: is poison ivy the only poisonous vine in Arkansas? Because I know you mentioned moonseed, but are there any other poisonous vines? I think with poison ivy, it's more of an allergic reaction to the urushiol oil that's on the plant, correct?
Yeah, exactly. So poison ivy is more of an allergen than an actual poison. It's just that we have that allergic response to people that develop that. So I'm just going to pull up the list of species real quick to think about that. Off the top of my head, the bottom line is if you drink too much water, it can kill you, right? I mean, too much of any one thing can be bad for you. And so as I'm working my way down through here and looking at the species again, some of the beans are known to have toxic alkaloids, like precatory vine has abrin in it, which is one of the two most deadly chemicals known to humans, right? So some of these things, yeah, you want to be really careful with. But then other beans we eat, the hops genus, right? Hops are these invasives. Well, there's a native form as well. Hops we obviously use for flavoring for beer and things like that. So as far as poison, nothing explicit comes to mind as a deadly toxic. The Gelsemium, the Carolina jessamine, I'm pretty sure I've read about that being poisonous to I can't remember if it's horses or cows, but certain types of livestock. So poisonous to humans versus poisonous to animals is often two very different things. The morning glories are known to have kind of again toxic chemicals in them that they can cause people use them commercially as drugs for hallucinating. So morning
glories, I believe, right? Like lysergic acid, adenine, or something similar to LSD.
Exactly. And so some of these, a lot of these milkweeds have alkaloids in them that are going to be very toxic as well. And part of the milkweed family is in the dogbane family. The dogbane family is called that. Why? Because it kills dogs. If a dog eats the milkweed, it might die from it. So yeah, a lot of these are going to have chemicals in them that you would not want to ingest in large amounts.
Yeah, and there are some, like the milkweeds, which I don't recommend foragers eat those, because they're very, the monarchs are highly dependent on it, and there's, we're really, they're suffering. But back in the 70s, before that was as much of an issue, you'd see that, in some of the foraging guides, like stalking the wild asparagus, that you could cook milkweed stalks and then eat those, and that breaks down some of that toxicity. But if you don't cook it enough or boil it in enough changes of water, it can make you very, very sick. So, I mean, some things are, poisonous raw or toxic raw, but, with cooking, it helps break down some of those compounds.
Yep.
But looks like Teresa Campbell recommends trying Baker Creek in Missouri to see if they have any muscadine. I do know they have a lot of heirloom, fruits and vegetables and whatnot. So, they may or may not have the straight species. I don't know without, checking with them. Razzmatazz, according to Vicky, is a hybrid seedless muscadine for sale in many nurseries. Let's see, Dr. Abbott, thank you for your organizational schemes. I think that will be very, very helpful. That's from David Darby. Vicky also says wild cucumber has little fruits that are okay when not too mature. I'm assuming that's okay to eat. But always do your own research, of course.
Yeah, I've eaten. I'm assuming that's Melothria pendula. But I've eaten those, and again, they're edible. They're just not. I didn't find them very tasty.
Right. Yeah, I've had quite a few edible things that I would say are not palatable as well. So, some people just refer to it as a wild taste to a lot of things in the foraging world, and some of those are compounds that we don't normally get in our diet and might be beneficial. Who knows? Ozark Soul, Missouri for muscadine. That's also from Sarah Gertz. They might have it as well. And Ozark Soul is going to be having a native plant sale in Eureka Springs coming up here later this month. I think you can check it out on their website or the Facebook page. Going out, they have the date on there. If you have any other questions, go ahead and enter them into the chat now. I'm not seeing any other at this time. But again, Dr. Abbott, this has been wonderful, wonderful presentation, a lot of great information, really packed and filled with all kinds of good info. And I know we'd like to have you back in 2022 to go into, maybe zoom in on some of these different areas or even have like a more, you and I discussed you maybe doing a series on just the foundational stuff. And I think that would be great as well. I think that would be well received. A lot of people would enjoy that. So just remind everyone, the recording will be uploaded to our YouTube channel. So check that out. I put a link in the chat box. But if you just go to YouTube and type in Arkansas Native Plant Society, you can view all of our past recordings of all of our webinars. But Dr. Abbott, we really appreciate you sharing your time and your expertise with us. I want to thank everyone else again for being here today. The next webinar is going to be on October 6th. That's a Wednesday at 11 a.m. Dr. Travis Marsico from the Arkansas State University in Jonesboro is going to talk about the important role that herbaria play in the recent discoveries of invasive species risks from hitchhiking propagules at international seaports. Again, our website is amps.org. You can go to amps.org/join and join right there on our website using your PayPal account. Check us out on Facebook at facebook.com/Arkansas Native Plant Society. And again, you're always welcome to send me an email at amps.president@gmail.com if you have any questions. Again, thank you, everybody. And also send me an email if you'd like a copy of the PowerPoint, or send Dr. Abbott an email. I'm sure he'll share it with you as well. Thank you, Doctor.
Okay, thank you. Thank you, everyone.